Okay. So for now you want to keep the...
Yeah, just some slack, just so we can cover the whole block.
Okay.
So he's moving north then, that should give us enough to turn a wee bit. Cover it, turn it, set, cover it, yeah. Yeah, just cover it, yeah. So we can go and so these things will be a lot.
I'm supposed to be nice.
Yeah.
Picking up all the time.
So, good morning everybody. This is Sergi and Anna-Helena greeting you from the Boytsov seamount along the Arctic mid-ocean ridge. Today we are at about 74 degrees north.
Can we start recording?
Yes, we can start recording. And we are at 2,400 meters depth, and the water temperature
We have just
is
started recording.
again minus 0.6 degrees.
Okay, copy that.
So we are back on the western side of the ridge, and we are on one of sort of 3 in a row known seamounts here at the southern part of the Knipovich Ridge. So when you saw that map in the very beginning, when you look at the bathymetry, the Arctic mid-ocean ridge makes sort of like an angle, almost a 90-degree angle, and we're now north of that angle, a little bit north, and everything north of that angle is the Knipovich Ridge until it goes into the Arctic Ocean and becomes the Guttel
Yeah,
Ridge.
sonar in a bit as well, Will.
Yes, please.
South of that, the one that goes more east-west, is the Moon's Ridge, where we have been earlier this cruise. So here we are on the highest of this three seamounts. So we have the Boytsov seamount, and now we have started at the bottom of the Boytsov seamount. So we are going to climb it. So we start now at around 2,400, and the top, which we hope to reach later today, is at 965 meters below sea level. Near us are also the Dibner seamount, which reaches 990 meters below sea level, and the Shigakov seamount, which also reaches 990 meters below sea level. So this reaches like 30 something meters higher than those two other seamounts. And all of these 3 seamounts are
The other side of the land.
relatively unknown, so this is going to be a nice, exciting dive for all, I hope. They were discovered in 1972. That's a long time ago.
For now we don't move the
But
vessel, just go away with the-
since then they haven't really been explored. So that's what we're doing today. A little bit of an unknown area.
Yeah.
And that's what we can find very often in the deep sea. Most of the deep sea is unknown, and
It's like
that's part of, that's where we're going now. Look at that beautiful pink amphipod swimming by. Are you looking at the sponges
Yeah.
already?
What else? Yeah, I saw the amphipod too. So it's quite muddy. We just started this dive, one of the deepest we had so far.
Yeah, and we are at the start of the mountain, so it's sort of rising up from the abyssal plain in many ways. And as you can see, the abyssal plain is also full of little life. It's maybe hard to see the big stunning secrets of it, but when we sample with sleds and stuff here, or box course, we find lots of beautiful things as well. So don't let this don't let this beige area just fool you. I think that was another jellyfish.
Yeah, it could be.
Clear it track.
Yeah, there's some stalked crinoids. Just
Yeah.
now, right in the middle of the lasers.
We are back in the stocked crinoids areas.
See, they have a similar color with the background. It's quite difficult to spot them.
But when you start seeing them, you see them all over because they're
You just got to
everywhere.
keep some in, buddy.
Yes, they are all over the place. These beautiful organisms, mostly from deeper waters. We're starting to see some rocks too, with our pretty well-known Lysodontorix complicata. This sponge that you see just beside the lasers, these spaghetti-like, right?
Yeah. And before we started the live stream today, I have to say Sergey already had his first sample of Lysodontorix. Because Sergey, as those of you who have followed us now, you know already that he is a Lysodontorix fan.
Yeah.
This is a species that is very widespread in this area, and that is strange because it's a brooder. So Sergey samples all of these to find the genetic distances between the different populations and to find out if there is a reason why they're so widespread, even though as brooders they can't really spread easily because they don't have larvae or anything like that floating around in the water.
The alignment of these rods seems to go in the direction of the water.
Yeah.
So we started right at the base of the mountain and we're immediately starting our climb. And I guess we're in for a lot of slightly sedimented hard bottom.
There's a crinoid in there, one of these free-living ones. There's these small sponges that you see.
Yeah, I was wondering what these white things are.
Yeah.
White.
For that we need
Are they the...
polio still
Yeah.
doubt. And this is an Exactinella. I think it's a Caulophacus sponge.
I put Exactinellid or glass sponge,
Yeah.
and there's a fish
Indeed.
with a dark head and a lighter tail.
We've seen a bunch of them already. Like.
Yeah, they are elegant. They're just standing there waving their tail, right? Like, well, here I am.
That thing is
Orange stuff in there.
They really know about it. Can't
I feel
Can you stop a
I think
second?
to also doing the same.
What do you think this is? Just a rock with this color?
It might be just a rock.
Yeah.
But I
maybe.
saw, yeah.
What do you think?
Well, I see amphipods hopping on the ground there, so I am happy. I think it's a rock though.
Yeah. Okay. Yeah, let's continue a little bit more. Sweet.
But we see this landscape is already here, sort of volcanic, right? Old volcanic. Look, and another
Okay, buddy, now I can stop when it's more favorable to you choosing.
Just for clearing the moments.
Oh, yeah.
I just because
Yeah.
from the side there, touch that.
Oh, yeah.
So we're going right next to the cliff, and we have to just move ourselves a little bit so we don't crash into it.
Good here?
Yeah, it's good. Do you just want me to continue up or?
Yes. Let's do that.
Yeah, there'll be other places where we can
Yes.
have a closer look at. So this region is like a really rocky, but there's also lots of sediment, fine sediment that are removed or like put it into what they call them when the ROV gets too close.
Yeah, then we need to use the thrusters a little bit to control that we are not crashing into the seawall. And then we disturb the sedimentation at that little point. And that's what we're seeing now. But we're slowly climbing up, and we're seeing that the cloud is going away.
Okay, if you need to go again, you can.
Okay.
So there's some
For now,
crinoids
we are
in there.
fine.
Yeah,
If you
lots
need,
of
I can just
crinoids.
rotate a little bit.
And, ooh, sea star.
It's
And
good for
just
you
to
rest.
remind you all
Yeah,
that
but
the
maybe
lasers, the two red dots, are exactly
to
10
stop.
centimeters apart. So that's how you can decide size of things here.
Mhm. So yesterday we were seeing that in these vertical walls there were like mainly these Geodia, potentially Geodia parva species, this round potato shape or with a greenish, brownish color that were carpeting almost everything. We don't find it now, these species, but we found, yeah.
So we do see a different fauna than yesterday.
Yeah, definitely.
Yesterday we didn't see almost any crinoids, and we had completely different, or not completely, but quite different sponge community. And here we have crinoids on every single edge,
Creep up another meter if you need to.
and
Just saw this Asconema megadriala, which is one of these huge jar-like sponges with browning color. Oh, you see here?
is that another? The
Yeah,
big
look
thing
at
there?
that.
The thing we have been calling the brown Asconema
Yeah, that one.
previously?
I think this is the Asconema megadriala.
Mega Atreala.
Okay, half a meter.
Oh yeah, beautiful.
Coming up.
Huge organism, that
Yes.
one. And in general, really big ones, the Asconemas that we found across all the dives. Some of these organisms can probably be like, I don't know, 200 years.
Yeah, because how slow do they grow really slow, right?
Really, really slow.
That's
Okay, half a meter.
the general thing here in the very
Can I
cold
do one
waters.
turn and off?
Yeah,
Take
go
a
slow.
bit more.
just go slow. Then slower.
We have very slow growth, and if we look at the
Yeah, that's good
size
speed.
of offspring, that is also quite often relatively large because they're relatively few. So those that make, if we compare them to more tropical species, the same groups here would be slightly larger, but grow super slow and become lots older and have fewer offspring.
Yeah.
So why are we visiting these seamounts that are unknown? So seamounts in general are listed as areas of vulnerable marine ecosystems because we do know that the seamounts, since they're sticking out of the abyssal plain, to some larger or smaller extent, they
Paddy, can you
are
turn off
these
the comms for the
areas of hard
Paddy, can you
bottom
turn off the comms for the live streaming?
that can support a lot of filtering animals, and these animals will tend to live more on, not only because it's hard bottom, but also because it rises off the abyssal plain, these seamounts, and they are sort of in the way of the general currents that are in the ocean, and so they create a lot of extra little eddies and curls and swirls and all of those things, so they get a quite interesting current system with the result of lots of extra access to the available food in the water. So very many filtering organisms will live on these hard bottom things that stick out of the ocean. So a seamount is always higher than 1,000 meters from where it starts at the abyssal plain and where the top is, and so at least if they're recognized. And this filtering sessile fauna, so fauna that just sits still in one place, are quite often making habitats for other fauna again. So these seamounts most often become like little oases, and that's not to say that the abyssal plain is a desert, because we've talked about that many times. The abyssal plain and the soft sediment is full of life, but it's a different faunistic habitat on these seamounts, and very, very rich and full of animals that, because they are needing to sit on one place to be sessile, they are quite vulnerable to things like bottom trawling and stuff.
Yeah, indeed.
And the thing about seamounts is that because they have all of this sessile fauna, also especially maybe on their tops, they are attracting a lot of fish and stuff. So from old time, before we knew that they were seamounts, a lot of fishers would know that there would be extra much fish around these areas. So there would be these places in the ocean where there would be a lot of fish. And we're now seeing quite often that these have seamounts under them.
Yeah.
So they're vulnerable to fishing activities. And through that are listed very often as vulnerable marine ecosystems.
So we're gonna do, once we get stable sampling, and we're just trying to get the...
Did you want to get another Lisodon
Yeah, we
Norix?
have one here and one here. With some stuff in here.
So Sergi is right on the sampling, and this is of course why we are here. We are here to study the marine biology in this area, to study the ecosystems and to understand them. Because if we don't study and understand them, there is no possibility of making informed choices for anybody.
Yeah.
We need to be able to tell the people who are making those choices what we need to give them proper scientific basis so that they can hopefully make some good choices, right?
Mm-hmm. It's not just identifying which is the fauna, which are the names of the different species that occur at different mathematical ranges and in different seamounts. We are also here to try to identify which are the connectivity patterns, how well connected, or if they're connected or they are isolated in between different seamounts, these species, and also across different mathematical ranges. And in order to do that, the only way to do that is just collecting a few samples. We are like, we have already identified one of these model species that is going to give us some of the ideas about these connectivity patterns. And this very species is Lisodendroris complicata, the one that you see right in the middle of the image, and the one that is going to be collected for further studies in the lab, further genetic studies in the lab.
And this will, of course, give us indications about how species in general can be connected between these seamounts. So we will do another, since we already did one sample before we started. This is the first sample of the live stream, and it's Serge's sample. Yeah,
and
the
a very
Lisodendroris.
big challenging one. So this one is going to be a slur. We have a tool that you will see in a minute coming to stage that is going to take this sample and it's going to place it inside one of these compartments so that we can later, when the ROV comes on deck, we can just later grab it and preserve it for scientific purposes.
And we've said this many times before, and it bears repeating, we have a wonderful team of ROV pilots here. So, because they are so great, I have full trust in us getting nice samples in difficult places.
Mm-hmm. It's actually almost vertical wall, so
they're just
the
flying,
they're
they're just flying in front of this thing, and look how stable we are. This is amazing.
Should we move to the tank one?
Tank one.
Yeah, first one.
It's not time. Even if you just sit away for it, I'll stick it on and then you go closer.
Yeah, there's no need to put in this one too close.
One out.
So we slurp it and slurp one.
Yeah. So you will see in a minute what I was referring to. We're gonna go a little bit closer.
Is it the this way?
The other one, yeah.
There.
Yeah, the spaghetti thing.
Yeah, that's it.
I see.
Open it a little bit
Why?
more.
If
Very close.
you want, I can push out the port drawer just to try and bring you back a bit, so just ease off your forwards.
Yeah, that's okay.
change it. Right, stand by our second tunnel.
Okay.
Try extend. Is that enough there? Just try and pitch down here. Come down. Aussie. Then you can reach Aussie left. Right up my shoulder, Aussie left. Just try and roll the elbow.
Here there is a...
No, not this one. The other one, the other one.
Yeah, we'll just try again. We're so close.
Yeah.
So it's on a vertical wall. I can bring it port a wee bit more if you want. Right, nearly there.
So the difficult with sampling when we're this close to the
Okay, ready?
when we're this close to a vertical wall is that the pilots don't have very much space to work with,
Yeah, like that.
and
Turn it
If you
on.
go backwards, you
it's
can
difficult to
Right, bring
pull
it port draw more.
the
Yeah.
manipulators this far back. They don't have the possibility like we have to pull the elbow behind us in a way.
Yeah, it's really challenging.
Yeah.
So we have to fly a little bit away
Now.
from the rock
Yeah,
so that
just
they
try
can
and
move.
use your roll there.
They can move
Shoulder
close to
fully
the
up. Use a
Lisodendroris.
roll and pitch.
Wait, I need to restart because this says error freeze.
Okay.
Okay, here we go.
Come up fully in the shoulder. So yeah, you're nearly there. Right?
Now, I think.
Right, you know, Über.
Now you think you're in position, right?
Yeah.
Okay, shout when you're ready. Left
Oh.
a bit. Just come back a little touch before you push down. Oh.
Yeah, well,
Easy, right? Easy, a little touch. Yep.
There we want.
Inside.
That's it.
Perfect.
Yeah, that's it. Yeah.
Slurp number 1, right?
Number 1, yeah.
Yeah, thanks. Antonio,
Yeah.
do you think you can reach? Think you can reach this
Jerky,
one?
yes, as well. So, jerky, yeah. So just one circle, just easy up there. Forwards.
Antonio, do you think you can reach this one, or is that too far?
2 seconds, I'll look. Where is it in your corner there?
Not to the right.
Yeah, right.
Yeah.
Now I'm there.
Yeah, it's too far. We'd have to move reposition.
Okay.
Right, you just gotta kick this door back now.
Okay.
Yeah.
Or else we can just go here. Do you think?
Forwards there?
Yeah. How many do you need from here?
Yeah, it's from here.
It was just to maximize.
Yeah. And you want them all in slurp
We
one?
just no. Yeah, then
Right.
definitely on the other slurp.
Mhm.
Want to go for the other one, Sergio?
Or this one back.
But then we have to move
Yeah.
horizontally further.
You think you can reach it from here?
Yeah,
No.
from here, no.
No.
Um,
Okay.
Yeah, you probably have to move a little bit. It's
Oh.
very close. The ROVs like.
Yeah, let's go
touch
somewhere
it.
else.
Yeah.
It's fine. Yeah, we can go.
So we will continue looking.
Yeah. Thank you so much, guys. That was challenging, right?
A beautiful sampling, thank you.
Welcome here.
Uh-huh.
Challenging.
It is challenging to sample on a completely vertical wall. We're just hanging there.
Mhm.
And flying or keeping the ROV just hanging like that in the water without moving is super impressive.
Any chance of a meter up?
Yeah, we should be able to get a bit of air, yeah. Okay, picking up slow. Nice and easy.
Thank you. And what we see is that the down here at still kind of the bottom of the seamount, even though we have been rising almost 80 meters already from
Yeah.
the start. That's kind of crazy. I have to double check that number. Yeah, we have been rising 70 meters since we started.
Mhm.
It's still sort of sedimented everything, right? So we're still almost connected to the soft.
Mhm. I
Um,
bet there's going to be a sedimentary area at the very
Yeah.
top.
that too. And this is of course sediments that come drizzling down through the area of the sea, drizzling down through the sea, landing on wherever.
And
But
yeah,
it also indicates that there aren't a lot of super fast currents around here,
mhm.
which is strange because we have all of these filtering animals still here.
I thought it was something different.
Did you check on the sponge again?
No, I thought it was like, how was this animal that we saw yesterday with this jelly that was attached?
Oh, the Lucinaria.
Yeah. I thought it was this.
Well, I think we saw one at the seafloor right before we started on
really?
on the mountain.
Oh, look at this green sponge. I wonder whether this is the one that Julio is looking after.
Julio is finding a lot of I would say, a lot of new species here.
Yeah.
Julio is looking at a lot of small encrusting sponges, and this is a group that has not been very well studied previously. And that does not mean it's easy to find these new species. It's just that there might be a lot of new species to discover. It's still a lot of work to find and describe new species, so
Yeah.
it's not to make Julio's work not sound big, because it is. And also he has the very good overview of what is existing already in the literature of described sponges. So
You may believe this is a low diversity habitat, but I mean, there's like at least 10 of these species that are megafauna organisms that are we've seen here. These endorix, the 2 or 3 esconemas, these white sponges, these encrusting sponges, these blue and green sponges. Crinoids. You see the crinoids in here? They're like in the crest. They're like
yeah, they're on
one
a
after
row.
the other. Yeah.
But it's interesting. Earlier we have seen them sitting on top of the sponges, and here they seem to like to be on their own.
Yep.
I mean, not on their own as in not around other crinoids, because they stand in groups or lines or whatever, but they're not standing on top of the sponges as much. But look, they're big and beautiful, aren't they?
Yeah. Indeed
They look
are.
like a sun.
And we're nearly at the top.
Of a small
You see? Oh, wow.
hill thing
This
here.
is the
Maybe now is the root.
yeah.
Oh, look at that's beautiful, huh? Look at all those crinoids.
Beautiful.
The line.
Especially when you... Can you stop here a second? Yeah, probably it's the difficult one.
And could we take away the lasers, please?
Yeah.
Maybe turn off the lasers?
This is off.
Yeah.
Thank you.
It's just to have this picture of
The three-dimensionality
here.
of everything, right? Because
Yeah.
right behind us is another wall.
just step back a little bit.
So we've come up this first little part of the hill.
This is indeed really beautiful. You can see the vertical walls that are just falling from this depth up to, I don't know, 2,000 or, yeah.
Well, right now
1,500.
we are at 2,350. And as I said, we started at 2,420.
Beautiful
So
one. So all these crinoids here, we will have a closer look at those. Let's make a close up.
I was not going to say anything, but is this?
Ah,
This
yeah,
is
these are very
endorix.
excellent. You see the detail of
Oh yeah, look at that.
these organisms. They're called sea lilies, right?
Yeah.
And they have multiple legs, or what do you call it? Legs.
Arms?
Arms, yeah. And each arm has like structures.
Extra spines to the side.
Yeah. And they are doing this to maximize the capture of the food.
And here you can also see in that one in the middle of the picture, you can see the structures that they're using to hold on to the surface below, so the hard surface.
These white
Yeah, this
expansions,
white.
right?
Yeah, it's like roots. It's of course not roots because it's not a plant; it's an animal. But they have these structures that they're using to hold on to the seafloor, and they can willfully let go when they don't think it's a good place to hang out anymore. And then they can use all their very many beautiful arms to swim.
Yeah. It's really beautiful when you see them.
Yeah, they're very
flying
graceful when
away.
they're swimming. So they are semi-sessile in a way, right? They're sitting for a long time in one place and then they move on to another place, if that's needed.
That was really beautiful. You see the particles coming from the very deep areas. They stay in there, so to grab these particles to feed on them.
Yay, cool.
Thank you
Should
so
we
much.
continue a little bit and we can maybe turn on the lasers again?
Now we follow this ridge on our left.
Yeah.
Yes, thank you.
Okay.
So we will continue our climb. Our main idea with this entire transect is to go up on the first little sort of pre-hill of the of the seamount. So we're going to stay in the around the base, but we are going to climb the lower parts of the seamount in this transect.
This is another of the
What's the change here?
species that's normally appearing in these habitats, the white barrel-like sponge that you see there. This is a Rosellida.
The Rosellida, we are back to the Rosellida. Yay.
Right? So Julio is with us.
Yeah, Julio came in to check
Can
when
probably
he's supposed to come back
Tell us
and
something about it.
spell us, but
Oh, this is
Now that
Bit
looks like an amphicella.
this one?
Yeah, it has a stem.
the stalk, the one.
Yeah,
with
yeah,
the
the
stalk.
stalk.
Oh, yeah.
So this is the one
can
we
see
call
it.
the tulip.
Yeah.
Yeah, we called it the tulip.
There are two tulips. This is the mysterious one
Really?
that our specialists in glass sponges don't know what it is. So
Okay, I am.
it's another tulip that is a little bit more robust.
Yeah. Sorry. The one that we collected before in our dive.
We collected my head at some point. Yeah.
So since
Mainly this one.
the expert calls it the mysterious
Yeah,
one, I am registering it as the mysterious one.
It is
Yeah.
better, no?
Another one there.
Oh yeah.
Looks like one of those chemistry balls.
Oh yeah. You see, this is the thing.
Like a
the
cauldron?
Sponge. Who are you referring
Could
to?
we call it the Harry
Hi there,
Potter
Antonio.
cauldron?
Hey, I'm here.
Okay. Yeah, I was trying to get your hand signals to stop me, that's fine.
Okay, guys, see you later.
Okay.
See you.
Yeah.
How much weight have you got here? Are you able to give us more after or not just now, just when we need it?
So Julio
I
and
can
Clancy.
give you, I
take
can pay
over.
you out a
In
couple
2
of
hours, in
minutes.
1.5
Yeah, we'll
hours.
stand by, we'll get Espen
You
covered.
are
and
still
then
stuck
we'll
with us for a
leave
little
in bit
while.
if you can. Not yet.
Okay.
Yeah, sure.
Julio
For
is
now,
in the lab
instead of
working
this,
up
Okay,
the
yeah, stay like
sponge
that, and then,
spicules
yeah,
from
sure.
previous dives and seeing if he can find even more new species. So we're never just sitting and well, as long as we are on shift, we're never just sitting and relaxing, I should say.
Here. So this is a beautiful one.
Yeah, that's a beautiful tulip sponge, right? And is this the mysterious or is this the not
I think this
mysterious?
is mysterious because it's like a
It looks like a cauldron.
yeah. You see
Or a
this
tulip.
stalk sponge?
Yeah, it's a beautiful one.
He's exacting elite. There's different groups of sponges. One of those is exacting elite glass sponges.
So you only need a couple of meters in.
Yeah, we'll just got
Those
one
are
little bit here. Stand by if
dominantly
you can.
Okay.
present
I
in
can pay in now.
deep
a couple of
waters.
Not yet, no, not yet.
No?
No.
Okay.
We'll just get Espen.
Beautiful
2
one.
seconds.
So this is like 30 centimeters from the stalk to the very top.
Okay, creep some in, just nice and slow.
Okay, just a couple of meters, okay?
Yeah, go for it.
That's
Go
pretty.
on, go on, go on.
But it does not have any pores on its stalk, right? The stalk
Mm.
is only there to get it off the seafloor.
Yeah, indeed.
So we
Okay,
see all the
hold on.
pores in the sponge itself.
Yeah, I'll try and get a wee bit
And the
here
big
then. I think I've only got a little wee
osculum.
patch. Yeah, a little wee
Yeah,
patch.
because
In
you're
the
at
middle.
very center.
Okay,
Yeah.
maybe
But
now
the stalk does not
we
have
should
these
keep it like
structures.
this because it's
So the
really
osculum.
tight.
Yeah, okay. Yeah. You stop coming up
collects
though, yeah?
all the
Yeah.
water after this
Very
water has
slowly.
been
Now
filtered.
we are filming one
using
sponge,
this,
so we are stopped with
Okay.
after this we can
chambers
come out
where they
again.
collect
Yeah, well,
the
I'll be sure once I finish.
bacteria and other food that gets inside the sponge, and then it expels the water through this hole, which is in the sponge jargon it's called oscillum.
And behind is more Lysidendorix.
Yeah, plenty of this.
Plenty, so we don't need to sample all in the first 10 minutes.
Yeah, we will try to get to a place where this it's easier for the pilots to land
Yeah.
their ROV.
So, what do you think those white balls are?
Okay, Antonio, if you can creep in
It's
a
a
little bit
question
more.
for Julio.
Oh, and we forgot to ask him.
Yeah.
He just
hold
left
on
us.
for a meter.
If you're sampling, I can do it here as long as enough to creep in.
No, but we don't have enough.
Oh, it's tight, is it?
Yeah,
And we
stay in
see
the same
some-
place.
Okay, stand by.
Stand by a
Some
little
of the
bit.
crinoids are actually sitting on top of the sponges because it's so full.
Yeah.
And, question, all of those red things,
And these are sepulets.
yes. When you just start looking at them, you see them, right? So Sergio's been so focused on all the sponges, he didn't see the serpules, huh? But look, the whole part of the mountain that is not covered by sponges is covered by serpulid worms.
Yeah, most of those I think they are
there
dead.
is swimming amphipods there.
Uh-huh.
This is
Yeah.
a nice little spot.
Yeah, we've seen this microhabitat, which is like a small reef built by skeletons of serpulids. They produce
Can we take off the lasers, please?
yeah. So in the middle of the image, you see like a small wormy-like brown structures. These structures are the result of the tubes, the remains of the tubes of these worms, that they make a substrate where other organisms like the sponges that you see can use as a place where to settle and become adults in there.
And then we see a crinoid cleaning some of its arms.
Yeah.
Maybe they are wanting to just get something
There's a
off.
bunch of different things in there.
Yeah. So this is what happens whenever we zoom in a place, right? We
Yeah.
see all these beautiful little details that we fly by when we are on our way.
Yeah, and it's one of the things that we are trying to do in order to describe these habitats and provide a list of the species and the communities
Yeah.
that they.
So let's fly on,
Yeah.
and
Yeah.
maybe the
Wait.
lasers again. Thank you.
Lasers on?
Yeah. So we will continue our rise up the mountain, up the lower parts of the mountain, and it's called Boytsov, and we haven't really found out why these three are called Boytsov, Dibna, and
So you'll be looking for information about the history.
I am trying Chigakov. So it seems to be named from three
Can
people,
play
maybe.
again, a couple of meters if you need.
Seems to be pretty common surnames.
Look,
This is amazing.
impressive.
Yeah, it's going straight up.
A little bit away from this, but you can
Dude, are you there?
identify how big this area is and how vertical. And despite it's vertical, it's been colonized by the sponges. You see this one with a really long stalk.
Yeah, and a shrimp sitting
Yeah.
under it.
Okay, dude, if you
Yeah.
need, I can give you 2 meters again.
Okay, stand by.
Okay.
Okay. Okay, Antonio, go for air, nice and slow.
Okay.
Another of these stalk sponges.
And then
in
we
the middle
have
of the
those
image.
grey and green encrusting sponges, right?
Yeah.
Yeah.
And Yeah, they're quite common. So once you see it, you identify that they are there. You can see that they are all over the place.
Okay, hold it there.
Yeah, that's the
Okay,
thing. You
we'll
just need to train your eyes to see things, and then you see them all over.
Yeah, just you will see now where the lasers are. This convoluted sponge. This is
Asconema foliatum.
foliata. Yeah, or foliatum. I cannot remember, but this is one of
On
the.
our list here, it actually says with UM,
Yeah,
but it's
quite common also. It's not the most common species that we're gonna find, but yeah.
it's still
And
very
in some
beautiful.
cases, you have this symbiotic life with amphipods, right?
Yeah,
Inside this sponge.
so this is just super crazy, dramatic landscape. And even though it's been mapped on a very general basis in 1972, it hasn't been super well mapped and published yet. So
Do you mean the community is living
no, even the
geology?
bathygraphy,
Okay, Antonio, if you've got more, just
Bathymetry.
creep some in.
Okay. thank you.
bathymetry and the geology not really well. We didn't find a lot of publications on this at all. But there are so many places to study, right? So
Yeah.
it's quite easy to find an
Okay, we'll stop there now.
Okay.
unexplored place in the deep sea, and that's one of the things that makes deep sea examinations interesting, is that?
Yeah, carnivorous sponge.
Yes, isn't it a carnivorous sponge?
Yeah.
Now, can we do a complete
Yeah,
turn
down
and
where
we
the,
stop
where the
again?
lasers are
Just
is
really
Yeah.
we happen to have
We are
better
passing through some big
places
rocks.
Would you need one more complete?
where to
Yeah.
look at them
Okay, stand by.
in detail.
Okay, yeah, go for it here.
Okay,
Pick up slowly.
coming in.
Yeah, I'll give you a shout when to stop.
Okay,
Or well, obviously you take what you need, but I'll tell you the area.
okay, okay.
How deep are we now?
Now
Maybe
we
I can
are at
one turn or a couple of turns and then we
2,337
stop again.
meters.
Yeah, no bother, that's fine, mate.
So I can see an Asconema megatriala.
Okay, I am clicking Asconema
Yeah, I think I'll look
megatriala.
good anywhere here, Antonio.
Huge.
Yeah, you carry on as
Huge
normal. I
tulip
think that's
again.
all
Is this a
covered.
mystic
Okay.
tulip?
So
Can we,
I
can we
can
just
stop now if you
Yeah, just carry on as normal, whatever you need. I think we've covered all the valuable there.
Okay.
Can
Okay,
you try
cheers,
to
mate.
stop there? Like I think there's a shelter. Yes, we would love to have a closer look at these tulip sponges.
Oh, but there is also one here.
Oh yeah.
If it's difficult to move, then we can just zoom in on the one in the picture
Which
here.
one? Say it again.
This one now.
There's two in there, and I think there's a shelter with a.
Come down there. Can you see where this one here?
Here, it's just.
Right.
But I mean, if you really we can land
Bridge,
here,
RV, can we stop for now?
you will see this is a really nice wall with those two tulip sponges.
Okay, thanks.
Are you going to want to sample taking it?
Maybe, but yeah, first it will be like a shot. So can you land here? This it.
You are on the limit. Tilt down, Sergio. You can tilt the camera down.
Is it steep? Yeah, yeah.
It is actually.
I thought it was not that steep.
Oh,
Now
it's here.
we can just stay here. If you can just stay still. Let's just wait for these to be clear. A little bit more.
It looks like it's all covered now, so.
Can you turn off the lasers, please? Thanks.
So we are standing here to look at the tulip sponges.
Yeah.
And are these the mystical ones?
I think that those are the rare ones.
Oh, okay. I like that some sponges are mystical, I have to say.
So is it gonna clear? Yeah.
Welcome back. And again we see all of these crinoids sitting on the wall together with these sponges, right? And
Yeah.
both along the edge and a little bit placed here and there on sponges, all of them, right? On those little round white balls. Ha. Maybe that's what I will do when I grow up. I'll stand around on a little white sponge.
The lights.
Okay, let's play a little bit with the lights and see if we can get... Yay! And we think the dust will land in not too long. And then we will have a clearer picture as well.
Yeah.
But this is again, you know, our pilots are making sure that the ROV is just hanging on this vertical wall. Yeah.
There's still a little bit of dust.
What should we have done without them? We couldn't have done a thing without
Yeah.
them.
Really big animals, these ones.
So are you thinking about sampling them or not?
Not yet.
No, because it's going to be
let's
super
just-
difficult.
Yeah,
Let's
yeah.
look at them and continue after
Yeah.
a while. It's a beautiful shot to look at. And ooh, I see it starts to clear up in that corner. So maybe it will start to clear.
Yeah. Yeah, there's a lot of sediment, fine sediment.
And we need to use the thrusters a little bit to hang here.
Yeah.
So we need to disturb the water current a little bit just to be able to hang here. But
It's cleaning now.
yeah.
This part seems to be.
The upper right corner seems to, or upper left even better, right?
Yeah.
Or maybe not.
It's taking a little bit of time.
So some of those Lysodendurics in the background seem to have thinner spaghetti than others,
Yeah.
as if they are spaghettini instead
Yeah.
of spaghetti.
We've seen that when collecting smaller specimens.
Is
with
it
the
because they are
one big
smaller
ones.
specimens,
Yeah,
or
I
is
think
it
so.
because they are a different group?
Yeah, that's a good question. And to answer this question, the only thing that can be done, or one of the things that can be done, is just study the spicules, which are the pieces of skeleton of
It's not really cleaning though, is it?
Yeah. Just continue with
Yeah,
it's
let's
not-
just
it's not
continue.
gonna clear.
Okay.
But thank you for hanging here.
Yeah.
This was beautiful.
Okay, thank you so much. So these are spicules. If you study them under the microscope, which is one of the things that
You will do
Julio
with all
and
of
Paco
the-
is doing. So they were comparing the small ones with the big ones. They came to the think that apparently they have the same types of spicules.
Okay, so that means that
Spicules.
it's the same species.
Yeah, it's just a matter of these organisms, the small ones, being smaller and having different
Okay.
sizes of the branches.
Can we turn on the lasers again, please?
Continue again, going straight
Turn on the
Yeah.
lasers.
We're just gonna swap pilots here, like
Yes,
Oh, yeah.
because now it's shift time.
I'm just holding this on. Yeah, put it forward through the wall, change it.
So, yeah. But could it be possible that two near species have the same sort of spicules?
Yeah, it could be. Yeah, because
Because
sponges.
there are only so many types of spicule shapes available, or isn't there?
Yeah, well, there are some sponges that they don't have just one type of spicules. There's some others that they have like different sizes and different shapes, and so there's a different combination of these spicules that make a sponge, a species of sponge to be identified as one sponge, and another one with another combination of spicules that you can identify as another species. But in this case, they have the same spicules, so it points to the fact that they might be same species. Oh, look, it's clear now.
But I was thinking, that's, I mean, if we compare all of the mammals, we have a lot of the same structures.
Yeah.
And
And you
you-
go to the genetics.
Yeah, right?
So this is the
Because
combination.
because you can't only describe on I guess this is what happened previously, that
Yeah.
we all only described on spicules, and now we're combining the spicules with the genetics.
So this is called the integrative taxonomy, right?
Yeah,
Where you combine
which we do on all different
the time.
groups now.
Yeah.
And on my amphipods, I have a lot of the same structures on all of them, and they are for me, it's the combination of a lot of things. So
Yeah.
I have to look at a lot of different, what we call, thank you,
Thanks. Oh,
morphological things
Yeah.
to be able to get to a species.
So it's a combination of morphological characters, genetic characters, but also behavioral characters. I mean, these sponges they may have or not these behavioral characters, but other species may have ecological characters. The distribution of the species, they can be really similar, but they can some species that just occur in a certain depth, or that they occur like in polar regions or something like that. So it's like a combination of information that helps you to identify different species.
So it's a little bit like detective work. Like I always think about it as detective work. I'm not
takes a
Sherlock
while, yeah.
Holmes, but it's a little bit, trying to find all the different clues and combining all the clues and finding out exactly
Yeah, indeed.
which species this is, and is it one, or is it two, or is it
There's
10?
a lot of cryptic speciation in marine habitats.
Yeah,
And then you have to be very careful when you're trying to describe a species, or you think that there's a new species, because you have to take a look at the previous studies that describe species from the same genus or same area.
and with cryptic species we mean species that look the same but are genetically different.
Indeed.
And they're difficult. They're very difficult to keep apart, right?
That makes
You
things
look at them and they look exactly the same, and then they have different genetics. And of course, it's still up to debate how large genetic differences are there within a species.
Yeah.
And this is not probably a thing that can be said, it's exactly this many base pairs of difference or whatever between two species, because it depends very much on the group, and also on, of course, your species concept.
Yeah.
And now we're getting philosophical, huh?
A little bit.
Yeah, but species concepts are so much fun.
Look at that huge sponge
Oh, you don't want to discuss species concepts?
No.
He's getting away from the species concepts. That's okay. We don't need to be too philosophical too early in the morning. It's stalked as well, right?
I think it's a Calophacus.
Calophacus, Arcticus.
Big, yeah. Sponge.
Did you see any amphipods sitting on it, or was it
Uh,
only the shrimp sitting on
I
it?
think. Well, I couldn't.
Yeah, the shrimp that we see here so far, those big pink things, they are Bithocaris leucopis,
Mm-hmm.
and we were thinking for some time that they were slightly different. You know, they had a little bit extra tooth there in the top of the head, or we worked a lot with that, and then we took genetics of tons of them to see. We were really thinking we were going to have lots of new species, and it's all the same species. So we really tried, but so what we did then was to show the morphological variations that can exist within a species, and that's of course the other way of crypticity, right? That you have one genetic group and they can have a lot of different morphological variabilities.
Yeah. There's a lot to be taken into account, because I mean you were saying this species definition. We've worked with sponges and closely related sponges in some cases, and just even tell which one is one because they're quite similar. They occur in different habitats, or in the same habitat, and we found a few examples of hybridization, which is when two relatively close species are able to interbreed in between them. So this makes everything more difficult. Right?
That messes up our general thinking about species concepts, right?
Yeah.
So that easy species concept that we learned at school isn't as, it might be a little bit too clear and simplified compared to how nature really is. But this is what makes it super interesting to study this,
Even more
right?
interesting than just the species that just...
See, he's hanging exactly like we were hanging earlier on this vertical wall. So
You see.
maybe that's who the pilots are learning from. These fishes were just hanging there.
I think that was the species, another individual for sure, that was being captured by one of these squids. Yesterday's end of our dive. There was
If you
squid.
didn't see that, after we're done with this live stream, go back and look at the end of yesterday's live stream, where we had a squid catching or having already caught fish.
Yeah, that was the best way of finishing to dive.
RB.
Well, probably not for the fish.
Probably for the squid though.
Okay, we are ready for the next move. Direction 255 at 0.1 continuous.
55 at 0.1 continuous.
So are the dives also listed in here with the depth difference we are supposed to cover?
How long it is? Maybe it's like we barely started.
Yeah, we have barely started and we have already covered 100 meters of height difference.
Can you bring the speed up to 0.2? Sorry.
0.2.
Okay,
And
thanks.
that is because in this dive we're supposed to put 50
600
so
meters.
almost 500 meters of depth difference in this first transect.
And how long is this transect?
Not super long, but we can see that the horizontal movement does not need to be very long for the vertical movement to be
700 meters this transect.
so 700 meters transect on 550 meters depth.
You can make the numbers, right?
Yeah.
Like all the time climbing the mountain.
So when we are looking at our transect here on the mapping screen, we see that we haven't moved very far horizontally, but we have, as we said, moved 100 meters up.
Yeah, we take the time to describe this community, different species.
But
different
this is
setup.
again a lot more sedimentary.
Yeah.
So we are on a place where there aren't that many currents and sediments get the time to land and hang around a little bit before they either are stuck or continue sliding down.
A couple of streams in the image,
The
easily spotted.
Bithocaris leucopis,
Mhm.
I would say. I can't, of course, see that from here, but I
Yeah, let's then.
can. I think maybe not the second one, but the one in the edge there is definitely.
Yeah.
Bithocaris has these really wide antennal plates. So when you see that they go a little bit in front of their head with large, wide antennal plates, in this area, it might be Bithocaris. And remember, most species are kind of, especially of these kind of animals, so small invertebrates are kind of local or
I think we
semi-local.
can stop here and sample.
Or you can check with the pilots if they feel like it.
Can you want these two? Now that it's a little bit more flat.
You think it's a little bit more flat?
Yeah. Is it?
Right in the edge, it's alright. Global manage.
See, we have the most wonderful pilots with us.
Yeah. It's always there's barely no time that they say I know if we.
And Sergi is just thinking that everything is possible now, because
Yeah.
whenever he asks about
I mean,
something,
it's
they
just-
say, oh, well, let's fix
This
it.
experience that I had so far, right? We have
Yeah.
had so far. They can make virtually anything.
Yeah, the impossible can be done.
So yeah, the idea is every time that we stop, we collect two at least organisms, so that to make the more of each of the landings.
Yeah, but when we land or hang in front of the wall like this, it's very difficult to use the.
Reach RV,
We
can
need to use
stop
the
for now?
slurp because we can't open the tray in the front.
Yeah.
That would take us too far away from the.
Just below you.
Wall. So, where do you want it in slurp 2?
More like two because
Is it slurp this one, is it?
Slurp.
Yeah. Just have to back off out.
One and one, the other one, the other one.
Yeah.
So then it goes
the
in.
one or
The one.
this one.
Yeah, and you want it in slurp 2.
Slurp 2 and slurp 3.
Okay.
Only with grease.
You seem to have more of it.
It's the funniest
So
part.
we're trying to position, we pilots are trying to position the ROV.
It's very interesting to see.
So that we can slurp it. We're gonna use this device that takes the sponge to each of the sponges to a different and separated place. What do you call that?
Hmm?
In each of the
Chambers?
compartment, these chambers.
Yeah.
So each of these ones are gonna go into a different chamber. So that we can later identify which individual was collected at each of the depths.
But do you still want them? I mean, if they are this close, you still want them in separate chambers?
Yeah, because they can break and then it's difficult to identify which one is the one. So this first one,
This goes to tank number 2, right?
yeah, absolutely.
He's ready to slap now yet?
Yeah, we are.
Okay,
Yeah.
give me those.
That's fine. That's fine. If we just leave some
Yeah. It's in the tank.
The other one in the other, another tank, another chamber.
One, yeah.
Different chamber?
Different one,
Okay.
please.
Okay. Ready?
Yeah. Hang on
Okay.
there.
Slurping.
So there was also a crinoid.
Yeah.
Good.
And then when you go.
Okay. Stop.
Yeah, thank you so much. So we can have a look around right now that it's clear. See.
Yeah, let's zoom in a little bit and look because this is a more sedimented.
We're gonna wait a second to go around. Yeah, before.
So
Where is that? This is Acceptinellit, probably. But I can't go farther than this.
but it has lots of, so it has crinoids and it's glass sponge.
Is there any amphipod that you see?
No, don't see them.
Those small things here, they are not amphipod.
Oh, there is an amphipod swimming,
Oh yeah.
and there is another one. Oh, and another one. They're all swimming around. Oh, look at
Seems
that.
to be hanging around this.
Yeah, maybe they want to.
Don't you think this is amphipods?
Maybe. Can we focus differently? If we, this is the focus, right? Focus. If we do like that, and then we, oops.
I'm not entirely sure.
Yeah, because look at all those swimmers, and they're sort of, are they eating from it, or? And there are other amphipods walking on the wall in the background there.
Yeah.
Oh, this is an amphipod heaven. Could we
Aha.
suck some of those amphipods into the same last, so
Yeah,
the
just zoom out a wee bit, show us where you are.
um,
On your port side there, okay.
yeah.
To the same chamber number 3.
Yes, we can
Same
keep
one.
it in chamber number 3. That would be lovely.
Okay.
You think this one? I mean, this is a huge one, right?
Yeah. Well,
So it's
huge
like
and huge.
Yeah, define huge, right?
But
You see
in this
this one
area,
door?
there is.
Yeah,
Yeah, but in this area, there is lots of amphipods. I mean, if we could slurp some of those, that would be lovely.
Not the sponge.
No, no,
Oh,
I'm just
yeah,
moving
yeah.
in slowly.
Yeah.
It's nice sucking, yeah, like that.
So I call it another cast, and we.
Yes, right. In between this, I mean, inside this crevice.
So again,
See them go now.
I think we just see this again and again. That
Yeah, give that a go, yeah.
when we zoom in, we see so many things, right?
Yeah, that's
See
one.
that.
Say what?
Okay.
Perfect.
A little more. You see they coming. No, it's fine. A bunch of them. I'm happy with it, I think.
Oh, I love amphipods. Yeah.
Stop.
Yeah.
Stop.
Let's have a look and see whether this white.
Yours light finished for today.
Oh, well, you know, I will never stop. So it's still there.
Still here.
No other in there.
Really?
He's hanging on, yeah.
Big one,
Shy one.
yeah. Yeah.
Let's see now if it's come. I think all of the small ones must have come.
Definitely
Or
there's a bunch
maybe
of them.
not all of them, but a lot
Just come
of, because
back
I
and
see
see what's there.
even more. It's still hanging there. Yeah. Maybe he deserves to hang there then.
Is that one right? Is it that one, or maybe something that resembles?
That's something else that resembles.
Otherwise, this is the strongest amphipod I've ever seen.
Oh, they can hold on. You know, of the strongest amphipods, I would say, are the whale lice. They are ectoparasites on whales.
The amphibod anymore. I think it's a sponge. Yeah.
Then we have all the amphipods, but really,
A bunch.
I mean, if you're an ectoparasite on a whale that dives down to 2,000 meters, you need to be strong to hold on, right?
yeah.
I would say they might be among the strongest amphipods.
So we have something else.
Thank you.
To keep you busy too.
Yes. Happy. It will be a long
It's
night.
just a matter of getting closer to.
Yes, it is all the
different
time,
habitats.
getting closer and looking in. So, from our examinations of this type of sand earlier, I would say that many of those white, tiny little dots in the sand are ball-shaped white foraminifera.
Yeah, indeed.
We have sorted through quite a lot of sand looking exactly like this.
And Jenny
yeah,
and Irina.
we all of us have been going through a lot of sand. There is an amphipoid swimming again.
You wish you
And
could
it's-
collect them, right?
Yeah. It's good to see that there are more of them, so we don't take out all of them, but we do take enough that we can study and see what the community is. That's our goal, right? We don't want to wipe out anything. We want to have a few specimens that we can examine and describe the community and understand the community, so that we can make or help decision makers to make good decisions.
Yep. Well, not too bad for now. How many casts?
We have 5 casts in a little bit more than an hour.
Okay.
Reach RV.
But we have half of our slurp areas full,
Bearing
so we need
0 at 0.2
out of 5.
Yeah, I
so
wouldn't
yeah.
do any more because there's the shift coming in less than an hour in 42
Yeah, they
minutes.
also need to have some space to sample. Right?
Look at this rock. Again, this
I wonder very
Tulip.
much. It's the tulips, all of them, right?
it's really beautiful. I'm not entirely sure we have seen this before.
So we call them the tulips again.
We've seen the other kind of tulip sponge, but not this one. I think. I may have not paid attention to this before, but
And almost carpets of crinoids. That's crazy. There are so many of them, and they are so pretty.
we are really interested in the most common organisms occurring in these areas, so that to make us an idea of which are the main builders, the engineers of these habitats, the species that commonly occur in these habitats, and just compare them with other similar areas in order to see whether these communities are similar or they differ.
It's near me. It's wrong.
Who said that?
The door. Probably the best way right now.
No. Here again, this entry area.
Is that the bridge just?
Yeah.
If it's,
Open water.
I don't know if it's really dark, but you make it.
Well, this is mainly sediment here. Do you want to do at some point? Let's just explore a little bit more.
Yes,
we could
but if
potentially
this
do-
is flat enough, maybe we could do
Swoosh
the swoosh course and stuff here.
So
That might
one
be a
thing
good idea.
is that we could potentially see a place where there's a rock with a sponge, and then sediment, and then we can trigger also the whisking bottles. Because we are after also trying to use an approach which is called environmental DNA, this environmental DNA, it's used to indirectly describe the communities, which are the species that are found in these communities. And in our case, we're trying to use 3 different approaches. So it's going to be push corers, meaning that we're going to get sediment, and out from the sediment we're going to be studying the DNA. So it's going to be also seawater, the DNA floating in the area, in the water column. It's going to be processed also in order to identify these species, and also sponges, since they are like pumping all the time.
They're like
filtering
filters,
all
right?
the time, natural filter feeders. So we're gonna try to also combine the 3 different approaches, so sediment, water, and sponges, for the communities to be described.
And environmental DNA is possible because all sorts of animals are leaving little pieces of them all the time. If you just consider us, for instance, we lose some hairs whenever we walk around, if we have hair still. We lose some skin cells all the time. If it's warm, we're sweating. If we have been drinking a lot, some of that water needs to come out again.
Yeah.
And all of these things that are leaving our bodies, and this is the same for all animals and plants and everything, we find DNA in that. And this is the leftover DNA that we can sort of check in the environment and connect it to the species that have left it. So it's almost a little bit like forensic science, right? It's a little bit like, ooh, there is a hair on this crime scene. Who can it come from? And then say we take this sediment sample and we check for the DNA, what is in there, and then we find out lots of different things. And to find out what those lots of different things are, we need to have a library that translates from a DNA sequence to a species. And that's another thing we're doing with all of the animals that we are collecting. We are building the library of DNA sequences. So without a local or relatively local library of sequences connected to species names, our environmental DNA samples can tell us that there are several things, maybe 50, maybe 82, who knows? But we need this library to translate those 82 something to 82 that we understand what it is. 15 sponges, 3 amphipod species, 2 worms, all of these things. And also to know which species specifically. That's where we need the, that's worm tubes, right?
Yeah, I think those are Sabellidae, probably. Because or probably those are the stalk crinoids. Difficult to see now because we have in the background has the same color as the. Yeah, there's definitely a life on top of this sedimentary area.
And everything that swims in the water, whenever they're shedding cells or changing exoskeleton or anything like that, all of this will land on the seafloor or hang out in the water, and this is why we can use water or filters the water or sediment to find out who have been in the area.
Yeah. And the beautiful thing about using sponges is that you can use the sponges that you collect here to identify the communities, but you can also use the sponges that someone collected a few years ago in the same area or in any area and just study which was the community at that point in this area.
Cool. How long time can that? How long can you wait? Because the DNA disintegrates after a while, right?
Oh yeah, but if you preserve the material well, like ethanol, and then you put it
In the cold and dark space.
cold, it
Yeah.
can last for a long period. Because in the end, what eDNA targets is small fragments.
And
Is
this is what
CO1,
happens to
18S.
DNA anyway. When it grows old, it fragments.
Egg fragments, but
Yeah.
since we're targeting like a relatively small fragments, the quality of the DNA is not that crucial in these cases. So like samples that were collected a while ago, preserved in 96% ethanol and put into the freezer fridge, they can potentially be good to do this approach.
So we're not talking about 100-year-old museum specimens, but we are talking about things that were sampled 10, 15 years ago, maybe?
Oh yeah, but even the ones that were preserved like a long time ago can be used as long as they were preserved in a way that DNA hasn't been damaged dramatically.
So not with formalin.
Not really. Well, for formaldehyde is a really good preservative. I mean, for getting the shape.
Yes, it's
Of
lovely,
the
and
animal.
it has been used quite a lot
Yeah.
in earlier times, especially because it was easier to travel with, and it's cheaper.
It is.
Yeah. So it's a quite common preservative, but it does not help the DNA as much as ethanol does.
It fragments a lot the DNA, but there's like different kits, genetic kits that you can use to extract DNA, small fragments of DNA, and then if you design primers like specific targets, which are like smaller, like 150 base pairs or something like that, you can still work on this information. It is more challenging.
We are growing into the super nerdy live stream today, huh?
Yeah. Okay. Yeah.
But that's
New
okay.
information.
Well, we are nerds, so kind of proud of it, right?
Yeah.
It's good to be a
absolutely.
nerd. And I guess at least some of you who are watching our live stream are also happy nerds. And that's great. Is that
Carnivore sponge, I think.
carnivorous sponge? A little lone tree.
Yeah. indeed.
Cool. That's beautiful. I was a little second thinking that those things on the side were not
Fish.
rocks, but
Fish, right?
yeah, they
resemble-
look like fish. Are
they
they?
are rocks pretending to be
No,
fish.
are they? Rocks pretending to be fish, or fish
Yeah.
pretending to be a rock? I think they're rocks pretending to be fish because they're overgrown by encrusting sponges. The grey ones, right?
Yeah. Now we had to zoom in so that to make sure they were actually. Oops.
We are still climbing this hill. And now we are at 2,280-ish meters.
Mhm.
And remember, we started at 2,480?
I think we can potentially.
2,430.
You think we can go faster? A little
Since
bit?
we are in the sandy area and it's
Yeah.
difficult to see all the beauty. Yeah, because it's so small.
Yeah. Well, you want a bit faster in here? A bit faster? Yeah. Please, thanks.
Sergi is interested in coming to the next exciting place.
Well,
I
we
think
just had a look
sponges
at
like
the-
this are kind of exciting as well.
Yeah, they are.
Rocks like this.
So my idea was to try to find one of these rocks with a sponge, crema sponge,
Mm-hmm.
and then use this sponge as a natural sampler and then pick the eDNA from the sample from the sediment and then fire the skins. But I'm not finding any small rock. You
Maybe
think
here.
This is a small
Yes,
rock?
but then we have to ask quickly if
Yeah.
it's possible.
Can you please stop a second? Sorry about that.
Sure.
Changing opinion all the time.
Yeah.
Look, this may be a,
Yeah.
can you go in front of that one?
On the way.
Because this is a rock, small rock, hopefully.
Can we stop the movement for now?
Or even there is a lizard under X, right?
Yeah, but we need a rock.
Yeah.
This one.
The problem is that we are needing a place where we can do the. Would it be possible to do push core here?
Push cool.
Is this a possible place?
Possibly, yeah.
Yeah.
Because then we could get all of the eDNA samples from one place, and that's the beauty we want.
Yeah.
Yeah.
Absolutely. And this rock can be easily picked up and
Yeah.
put into one of these compartments.
So we will first do push cores,
Yeah.
and then we will do Niskin bottles or water sampling bottles.
So yeah, you will see
yeah.
which is the procedure. So it's push core number one.
Yeah.
So,
We can, if it's possible, we can do both of the push cores here,
yeah,
sort of next to each other,
two replicates.
not on each other, but
Two pushing.
Is it fine here? Harry?
Yeah, it's really good.
we used to have half an hour.
Half.
Yes, 30 minutes.
You can wait to take over, no?
Julio is very eager to take over, but he is not super interested in the push course, so we can do that first. And I will write down all the details for the sampling place.
They're wrong.
Yeah. You gonna do the mugs too?
Oh, we can do one mug here, maybe.
Yeah, definitely. Can you turn off the lasers, please? Lasers off.
That's as far as I can go. Yeah.
That is a beautiful push core.
Indeed.
Zoom in. Good.
Yeah, good job.
Zoom in a touch, Joe. I can see the top.
Beautiful. Thank you so much. That was one of the push cores to come.
Oh, you know what? I have written this wrong.
nice.
Zoom.
Backwards.
Field of depth is a hard thing to see when you only see things on video.
Mhm.
Could we please fire the two Niskins as well?
Push down, is it?
So Paddy's making sure that the two bars
Yeah.
like
Zoom in's okay?
yeah.
We want them to really stick in the holders. That's beautiful. Thank you.
Good job.
Is that everything with you, Aaron?
No, if we can pick the sponge. Do you want to pick it or do you want to scoop it?
Where is it? Zoom in.
Pick it. Or if we scoop, and then it should
Can
be
you
about
zoom in
one.
on it?
It's pretty small, right? I think it got broken.
Yes, this rock right in front of. No, actually, it's right in front of the. No, it's further in.
Top
Down? No, down.
That's here.
There.
The thing is that it's broken now. Oh yeah.
Yeah, so it's the one right
Let
in
me
front of
check.
the.
Let me
Oh,
check
the
1
tray.
second, buddy. I'm gonna look whether there's any other. Is it that one?
It's not on a rock.
no.
So you need to take the one that is right in front of the tray.
Yeah. Can you go for this one, buddy?
Yeah, how are we going to pick it though? Is it really attached to the rock?
I think it's a loose rock. Hopefully.
That makes it a little bit more harder.
You wanna scoop it?
Yeah, we can try, yeah. We can try scoop.
Okay, so then we do that before we do.
Antonio tried before with the arm, and it was like a pretty one with similar rock size. But I mean, up to you.
No, I won't scoop because I'll be digging the arm into the sand.
Yeah.
So then we are done sampling the sediment itself, and now we are off
Scoop, right?
to
You want the scoop? I scoop. The mud.
yes, but first we want to sample
Yeah.
your scoop up your rock with the
Yeah.
with the sponge, so that you can use that sponge as a natural sampler in a way. Not only in a way, properly as a
Yeah.
natural sampler. And we want it to be relatively close to where we have the sediment samples, so that it's very possible to compare things.
Yeah, indeed.
Yeah, try to do this as by the book as possible.
Awesome. So in this case, we're going to use a scoop in order to get the whole thing.
Zoom in. Set up your camera and zoom in. Yeah, zoom.
More zoom?
Yeah, yeah.
No, I think you're right to the left side of it in a way.
Oops, that's hard this one.
Grade me in there if you tell me what you want.
Bring the pitch back.
And a little bit more to the right.
To the right.
I'm
Yeah.
up against the stone. See the mesh? See the holes in the scoop? They're right parallel to the stone.
Yeah,
Sort
but
of
idea
on the
was
side of the
to
stone
go to
instead of-
the stone.
You want to take the stone as well?
I think it's too big.
No. Pushing your way away.
Yeah.
If it's so big that we're pushing the ROV back instead of having the stone, then it's too big stone.
Yeah, don't worry about that.
So I'm trying to scrape it off.
Okay.
We zoom in. Tilt up, zoom in.
Don't worry about it. I thought it was like loose.
Pushing it up, pushing it like it's solid.
It's fine.
Will I try scrape?
No, can you then scoop one of the big ones or
But scrape that because it's going to be, you know, those others are stuck on the mountain. But maybe we can scrape those off.
or scoop?
Where do you want me to go?
Yes, right
Where
there.
the big ones are.
So a little bit further back on, yeah, if you zoom in on that one of those.
Yeah, okay, let's see what we can do here. Tilt up. Tilt up the camera. No. Zoom in.
Like this?
And zoom in, yes, perfect.
Yeah. I don't know. That's a difficult one.
Am I in full stretch, Rob?
Sorry?
Am I in full stretch?
I don't think so. Not yet. No, you're not full stretch.
Okay.
You've got those to go, yeah. Pushing.
So sometimes sampling is quite much more difficult than it looks because manipulating these arms in an
I
environment
think it's
where
better if you scoop it,
we don't even
slope
know the
Correct.
size of
isn't it?
the rock they're sitting
I'll
on.
be able to reposition for that to start with. Zoom in.
It's quite difficult sometimes, huh?
I think it's like this, not in a flat
Oh
position.
yeah, but we are touching it. Maybe we can scrape it in.
No.
Okay.
Fine.
It's the ledge on the bottom right is catching it. Let's just leave that there for a moment.
Yep.
Yeah. Is it possible to fire the Niskin bottles while we're thinking?
It has all this sediment around them.
Oh yeah,
You
so we will
want-
wait a little bit until the sediment has landed. That's a very good idea.
Open it around the camera.
Can I try?
Yeah, just pan around.
Later we can use this one.
This is big
one.
just for how
For-
slurp or pick.
Yeah.
Which
Pick, I think.
That one, big one.
Can you
It's a
just
big
pick
one,
it
can
with
just
your
pick it on
hand?
the very.
yeah. Okay.
We will try, but for now, thing is, we do this, the mug,
On the mug.
the mud, right?
Right, so I put the no slurp. So
No slurp.
No, I'm gonna put away the put away the
No slurp.
put away the
Yeah, the scoop.
scoop.
Mm-mm.
Zoom target.
Sure. So this sampling is gonna keep us busy for a while, right?
Yeah, it is.
I think here the mug
Yes.
is closer to the just one replicating the
Yes.
final one. Yeah, thanks, nice.
So
Any of the maps that you would prefer?
I would prefer resumption easier.
Yeah.
Easier.
So then you can probably explain which is the purpose of doing this.
oh yes,
inlet.
it's not the purpose of drinking coffee, even though of course we do love coffee. And they have good coffee machines on board here.
Sure.
But this is to get a part of the sediment, because as we have talked a lot about, there are a lot of animals living in this upper part of the sediment, and so this thing that looks like a coffee mug without a bottom, so a bottomless coffee mug, that's kind of cool. But it has a mesh at the bottom there instead, so that we gather up, we use it to pick up sediment, as you can see, and then we bring back
Oh, that's a full one.
a nice full sediment sample back to the lab that we can use some hours to clean, and then some more hours to pick through and pick out all of the small beautiful animals that live in there. And when I say small, I mean really small. It's like some of them that I picked this morning are an average, I would say 2 millimeters long.
Really small then.
Yeah. So you need to develop an eye for that, of course, but there is a lot of biodiversity in the sediment as well. And to make sure that we don't forget to sample this, we bring coffee mugs along. And then we can think about drinking coffee as well, right?
Yeah, probably we're gonna need it.
Yeah.
After waking up so early today.
Ooh, but I'm a tea drinker. On land I drink coffee, but at sea I drink tea.
Why is that?
I think it started when I started going on cruises like this, and that's so long ago. We're not talking about
Stuff
that.
done
But
into the net, is there?
I think so.
yes,
Still sitting on top.
Plenty.
and the coffee machines were not as good as they are now, and the coffee was kind of disgusting. So I just didn't want to drink coffee, and then it just became a thing. I just don't drink coffee on vessels anymore. Even though I would have to say on Norwegian vessels, most people drink coffee all the time. There is a lot of also in the mug. I think this is going to be good.
Yeah, plenty.
10 minutes until tape change.
Okay.
How does it look around the Niskins now? Is it still muddy around the Niskins?
Definitely in the water.
Yeah.
I'll give you one camera so you can sit there.
So if you can. Number one, quad one.
So since we have to move to collect these samples, maybe
Yeah. Maybe if we move a little bit to collect your sponge samples, we can do the Niskins
don't worry,
there.
Andrea, because we're gonna move up a little bit.
Just for you to look on this camera, on
Yeah.
this slated monitor.
Mm.
The first one is where the Niskins are.
Yeah, you can see now. Oops.
But we wanted to, when we're sampling, we wanted to sample the width of the community. So we're focusing a lot on the sponges when we're talking and when we are sampling, but sponges are, as we have said many times, also habitat for a lot of other animals. So when we look at the sponges, we also find the community. And then to get the infaunal community, so inside the benthos, in benthos, we do coffee mugs.
Okay, that's it for sampling for the moment.
Yeah, we're gonna move,
Like maybe 3 meters.
yeah, somewhere around here to pick this big one, and then we'll fire the liskins.
If the water is clean.
Yeah,
Yeah.
Okay. All right, Rob, yeah.
and then we'll complete this sampling,
Yeah.
which is a good one. That's gonna be a pretty nice one.
Yeah. So we are going to sample this one, and then we're going to
This one
fire.
almost.
Some line there, on the edge.
Yeah.
Yeah.
Here.
The supply line there, I'll be on solid.
Yeah.
Okay.
Well, if you, I think
Okay,
you
let's-
know
let's
exactly
start
what
with
you
the
need to do.
Oh yeah, the one that you prefer the most.
So we have to know where we're going to put the sample then.
In
One of the right throwers.
Yeah, but if the RV's pinned up against a vertical wall, how do we push out the drawers?
no, then you can't.
No, we can do it.
Or you can do everything.
But it's not a, click the fingers. It just gives a little time to settle for me.
Yeah.
Yes. There is still space, a lot space in both drawers. So
Okay.
whichever drawer is best
More
for
than
you.
likely, yeah, port.
Okay.
Yeah. Yeah, no worries. We will take the sponge, the liskins, and so there's this. We can just wait. You already have one, right? Same one, the one with the okay.
But we got that one.
In the bio boxes, we have coffee cup there, and we have two Lysidendorics there. But in the slurps, we are a little bit more packed. So we have Lysidendorics in 1, 2, and 3, and in 3 we also have amphipora.
Okay, nice.
We had a wall of amphipora. That was crazy fun. Yeah. So you have 2 slurps and plenty of space in the bio boxes. But remember that people coming after us are also going to be allowed to sample.
Yeah.
Yep.
And we're at the beginning of the second transect.
Oh, we are still at the first transect. This is
Jewish.
This is the end of the first.
Ah, it is. Okay. But it's...
Is it begin? So sorry, we're just going to pick this with the jaws, yeah?
yes.
I think so.
Please.
Yeah, you can.
5 minutes until tape change. But we are,
Are you okay to start sampling?
yes.
Yeah, thanks.
We have been going 150 meters up, but it's 550 meters up, so there is lots still to go up.
Yeah.
Maybe you can check upstairs.
Oh, there is a beautiful little shrimp behind there.
Yes. And another spot here. I don't know.
Yeah. No, that's another shrimp.
Sorry.
It's right there. So, Julio, we have been discussing. Have you looked? There were all of these white little balls of sponge.
Yeah.
So that's a thing that maybe you can explain to all of the people watching, because we couldn't.
Well, you have many species actually.
Like these white little balls there.
Those are potentially little polymastia,
Front right.
but it's a really diverse community. So we have polymastia, we have
Yeah, rear right.
what we think it's a new species, has to be here. I believe it's here.
BB4?
For that I will need to look into
Yeah.
more detail. But collecting like that is very useful. So collecting always a piece of rock is great, because
Yeah.
then we can check those little
Awesome.
It's 47. And we're slightly
And then we
over.
need the knee skins. How
Okay,
is it?
so we finished
It's
with you
kind
now?
of,
Yeah,
yes,
thanks.
please.
Awesome. It's quite clear, right, with the knee skins?
Firing the bottom one.
Yeah.
Okay.
First.
Start. Top one, too? Do you want both?
Yeah.
Sorry?
Yeah, just in case. Thanks, Andrea.
Okay. Closed.
Both, okay.
Both.
Thank you so much. Great. So we collected also the 2 knee skin bottles at this point. For the eDNA from the seawater. So these bottles, knee skin bottles, are ready.
And we placed it in BB4, right? And that was cast number 9.
Yeah. So can you put on the lasers, Rob? Lasers, Rob? Lasers. Lasers. Yeah, sorry. Awesome.
Okay, and we have approximately 1 minute until
1 minute and then we'll
tape
change.
change.
Yeah. I mean, take your time.
So, yes, and after the tape change, Anna-Helena and Sergi will go and get some lunch, and you will have Julio and Franzi here to take you through the next part of the dive, and maybe you will see more of the walls. We've had quite a lot of beautiful walls until now. So, tape change in 15 seconds.
One, two, three. Stop recording.
So we take the tape to the dry lab. Do we need to tell anyone in the dry lab that?
Yes.
Could you please tell the dry lab that we changed tape? Sorry. Tell the dry lab that we changed.
Oh yeah. Dry lab RV.
Dry lab.
Yeah, media has changed. So what you saying?
have to.
Yeah.
So what way did you turn?
Has to go counter
Return to port, didn't we?
No, we turn to starboard. Yeah. Okay, 1, 2, 3. Recording.
Okay, so that's it for us. The boat is moving a little bit as well today, which is
That's where the fun is
interesting.
when you're trying to sample.
Yeah.
It feels
Yes,
like you're going down to it.
because we don't have a lot of waves, but we do have a little bit of swell today, which is impossible to feel. Oh, thank you.
So you kind of what you did, you took the turn down.
Just need to stand up in all of this after sitting for 2 hours.
Yeah, that's the easiest.
Enjoy lunch.
Okay.
Thank you.
There you go. Yeah, it's coming in there now. The ball is there. Up to.
Thank you.
All right.
Something.
No problem. Thank you.
Okay, so we have 5 sometimes 6. Too much.
Cheers, no worries.
Hello.
Hello. Julio and Francia are
Adios.
taking you through the next parts of the diet.
Oh,
It's always good. So what's going on?
so we're still at 2,270.
Oh, sorry.
Do you remember, Fancy, how shallow or how deep was the end? of this?
500
No, the end.
at the end.
That's the beginning.
Let me check the wise book.
Yeah. So this should be like climbing a mountain. Like this.
You said the end? 1,885.
Okay.
So we still have 400 meters
Yeah.
to go up.
So that's an Asconema, Foliatum.
I'm logging.
Yeah, plenty of the Chalendorid. So this is the off-rent community with the Chalendorids and crusts, Medesmia, Alisarca probably, Laniera, all that stuff. So it's quite different than the communities of yesterday
Yeah.
on the unnamed.
We're also a bit deeper at the moment,
Where people, yeah, you're
but
right.
we also changed our position to a site that is again more to the south at the Knipovich Ridge, but closer to the Mons Ridge, where the two ridges meet, and we're more on the western side again, in comparison to the unnamed seamount where we were yesterday at the eastern side.
I'm thinking we should at some point collect one of these little rocks
With the round sponges on them,
for the yeah,
and the uncrusting sponges as well.
yeah. No, for the encrustings, but maybe we can take a roll with these Arendorids and some grass. Let's see.
Yeah.
It's a little bit steep down here.
It is. Yeah. So we're going up this wall of this seamount called Boytsov. We're now more on the southeastern side of the seamount. Yeah, we're at the steepest part, I guess, at the moment. Here, look, we're going up this spar to a little plateau, and then it's also going up, but less steep.
So what makes the seamount different to the other ones that we've been before on the, let's say, on the western part of the rift, is that this is not facing immediately after the rift, but there are two other seamounts in between the rift and this seamount. while both Boyd and Schulz Bank were right in front of the rift. Well, right in front. They didn't have an extra mountain system in between.
So do you think that that does what to the community? That they have more mountains? What does it do to the communities that this is less exposed, could you say? Or
well, I'm not sure. But, well, actually it looks very similar. So I'm missing the fan-shaped sponges. You remember?
Yeah.
Stellodorids. Yeah, maybe that is one. I don't want to move the camera that much, but that looks maybe like a Stellodorids. I was just saying that Stellodorids is not here. But in the other dives, they were very diverse. They were creating like little gardens.
and there was this
I don't know what is that.
on a stem. It's also unknown sponge. Unidentified sponge.
Yeah. One of many.
It's really different that the ship is now moving again. I have to get used to it again.
And I guess it's different. It's difficult for the rock diver as well.
Yeah.
Because so the umbilical goes direct to the vessel. They feel the movement of the waves.
Yeah, we were really blessed with super flat water, no swell whatsoever for what feels like a very long time. And now we have some swell coming up again and a bit more movement.
Is this easy to land here?
It's uphill. Do you want to look?
If just to look, we can do that.
To collect one of those little rocks, but there's no rush.
Okay, let's try and get something less steep if you can.
Yeah.
I think we'll find more rocks here.
Can we start
Is
moving
the fish look very silvery, or is it just because of the light?
0 at 0.1, please. Sorry, did you heard? Bearing 270 at 0.1.
Yeah, I think it's a light.
Yeah,
Thank
it shows
you.
a very upright sleeping position.
Come forward around.
Oh,
Something going on here.
Big rock.
It's rock. It's a cave.
yeah. Lots of crinoids enjoying the currents. Acidian and encrusting sponges there. Little amphipod swimming by.
Where?
Or was it a little shrimp?
So the anemones?
No, yeah, I didn't see them yet.
I'm not sure they saw anemones in this dive before. Sergi and Ellen.
And a lot of these tube worms.
Yeah. And Asconema. Megatriada.
Yes.
Potentially. Asconema foliatum. More Lisodendorids. And see how many grass are there.
Speed is the ship moving.
These crusts.
I think they have seen them before.
I think we did. But some of them just looked a little bit more brownish, and I was wondering, like this now looks again whitish.
Welcome to my world. Uncertainty. Yeah, I mean they look like the one that you and me collected some days ago with the stud gun, but it's very hard to say.
Yeah.
We'll be looking for a rock with grass. The more grass, the better, and the smaller the rock, the better. and the flat the area, the better, so they can stop and collect. So that's what I'm looking for now. It's nice here, all the crinoids on the edge. Let's see.
And there's a big, no, is it a big
It's a carnivore
sponge?
Yeah. Okay, I think we can move forward.
Yeah. Mm-hmm. Yeah, let's go on. Yeah.
Oh, around it anyway.
Thank you. Sponge that you mentioned before.
Sorry?
No, I just pointed the carnivore sponge that you mentioned before, Avistocoma.
7 days ahead.
So this is a really
Off
big
into
rock.
the blue. Oh.
Show the arm a little bit.
Avistocoma.
Wo ist es? Was kann nehmen? You don't trick me anymore here.
Now, Scanema for Megatriada. And plenty of
Plenty
little
little-
sponges and Lysoendorids. So Lysoendorids is the queen
Yeah.
of the banana hole.
And even beyond that.
And even beyond, yeah. Taking over.
Yeah. It's not. Yeah. So the banana hole was the area of international waters that we've been in before. For example, for the dives at Louisa Boyd, and at the moment we left this so-called banana hole, which has its name because of its banana-shaped outline when looked at from beyond, above on the map. And we're at the moment on seabed that is claimed by Norway, and also the water column is in the so-called territorial waters.
Oh, check the green one. You remember the green crust?
Yeah,
I think it's here.
we sampled the green crust as well at one point.
Yeah, well, we sampled something green. Yeah, probably the same, I would say.
And there's it's a crinoid on top. Do you have a name for the green crust already, or it's just green crust? For
It's
now?
Nimedesmia.
Hemidesmia.
Yes.
Hemidesmia.
There are many Nimedesmias in the world. It's a super diverse group. That's why it's a little bit difficult to know the species name.
I have the feeling all sponge groups are super diverse, or are they? Some are mega diverse, I guess.
Yeah. Well, some groups are not that much, but most of them are. Yeah. I mean, they had plenty of time to become diverse. They appeared more than 500 million years ago.
Making them presumably the oldest animal on Earth.
Yes. Or potentially,
Yeah.
I'm not sure.
Yeah.
Yeah. Now the discussion is whether they are super old or even a little bit more.
No. And are there some sponge species of which you know that they are especially old, or they look still very similar to what they looked like millions of years ago by fossils or some analyses that you could do? Asking tricky questions here.
I'm not sure. Probably there is an answer for that. Because I do believe that there is a calcareous sponge that has some affinities with a group of sponges that are very common in the fossil record. But yeah, there's not much I can tell you about that because I never really dig into that.
Yeah.
But all of them are quite old-like to me. Like very, yeah. Like when you think about a very old landscape, you're thinking things that look like a sponge, right? That are not very, without the defined shape, without true tissues, without symmetry. All those things were invented later in the history of life.
Can we zoom in on the sediment a bit to see what it's?
I just want to check that one.
Okay.
Because it looks like a painting. or
Yeah.
something.
Beautiful. 3 sponge crusts of different colors.
Yeah.
Ah. And here's a long. No, it's a crinoid, I think. No, I'm missing the top of it on this little stem. Oh.
So here we can see the diversity. So all these 3 things are different. You can tell by the morphology of the holes, by the color, by the texture, for sure by the genetics. That's
All competing for the space and
wanted to see
they're all.
wanted to see
Oh no,
something.
there was I think a large. Yeah. Something on a stem.
It was like a crinoid.
Yeah, like a stalked crinoid, I think. But I couldn't really see the top of it, but I think it was just because the sediment hid it a bit.
Crinoids, the shrimp.
Yeah. And there are some bigger round sponges.
Yes. But it's really hard to tell without having the sample. Look at that one.
Yeah.
So, right, you see.
But we see quite nicely here that all the space that is not sedimented, that is still rock substrate, is populated by mostly sponges, either uncrusting sponges or sponges that grow in other forms. But yeah, this rock surface is really popular.
Can we change the bearing again to 260, please?
Here's also coming up in the top left corner a stalked sponge, one of the tulip-like sponges.
Yeah.
Even more of them.
Looks like an amphistella. This is on the list.
Is it?
I think so.
Ah, like this, Monai.
Yeah, I would like
Presumably
to check
Monet.
that one because
They have a nice
that's-
fan over there. Or no,
this one,
I haven't
or if
seen that
it's-
for a while.
If it's Hemigelius. Okay.
Was that the one that had this more transparent
Hemigelius.
outline?
Hemigelius is one that, if you remember, we saw it with little triangular buds on the edges. And sometimes it has a little bit of a color, like orangish or yellowish.
Oh, we see some fish here.
So we have done about 200 meters of transit.
Just going further uphill here, and we're at 2,200 something depth. Meaning we already covered 200 meters. Yeah.
Can we increase
200.
the speed to 0.2, please?
Also, not only length but also height.
Yeah.
That's what you meant in the first place.
Well, it's probably more than 200 because it's also up, so 200 in horizontal.
No, probably not. Or just about
Okay.
to get there.
So how does it look like? Okay.
And we just climbed this.
Yeah, that was the rock,
Yeah.
rocky area. Okay.
Somewhere maybe here. And now we're going, the slope is sloping.
The slope is sloping. So we still have 400 meters to climb up until the end of the transit. I wonder if there is a ridge at the very top, as we have seen in some other dives, ridge with all the filter fadings lined.
We're here to find out.
Yeah, that would be something. Because then the second transit follows that ridge. So if that
that would be
happens,
cool. Yeah.
then it will be. Right?
Yeah, so we're
Yeah.
climbing up the
Exactly.
slope to the ridge. Then we're following the ridge. and in comparison to the slopes that we crawled up yesterday and the day before, now we have, which were very rocky, we now have more of a sedimented slope.
Sedimentary.
Yeah. You see still some rocks out, but a lot of them seem to have been buried. And this darker shape, could this be our foraminifera again?
Looks like very much to me. Yes.
Yeah.
So there's a bunch of them.
Yeah, so little organisms agglutinated.
Yeah, well, Foraminifera
Yeah.
is single-celled.
Yeah.
But I think that those are maybe together.
Yeah.
Several individuals. A good friend of mine studies the Foraminifera. They were always very present.
Is that friend watching maybe?
I don't know.
If so, please comment. Greetings to Julia.
255, that's the translation. 3 Tel-Aviv. Can we change the bearing to 255?
255.
What are the little
Thank you.
darkish? Yeah.
That looks like a rock.
Yeah.
Yeah, two rocks colonized by little sponges.
Ah, yeah, that's it. Of course, they're colonized.
Maybe we can speed up a little bit.
Okay.
Fancy? What do you think?
Mm-hmm.
There's a fish over there.
On the right side?
A small one.
Yeah.
Not much to say then.
Yeah, that's perfect.
That good?
Yes, that's good.
3 Tel-Aviv.
Here also probably just a little rock that is encrusted already.
To
Yeah.
0.3, please.
36.3.
Thank you.
And the dark patches are all little rock pieces? Or could some of them, like yesterday, we also had some dark anemones.
This one here?
Yeah, just wondering if it's all rock pieces.
So the two lasers are 10 centimeters, right? That's it.
Yes. What do you
Hello,
have?
check Trilab.
Go ahead, Trilab.
Just a heads up that at 2 o'clock we will break this line and go to the school stream for an hour.
Okay, copy.
It happened to me that every time that the samples are on deck, they are bigger than what I thought.
No. Oh.
Maybe it's me, maybe. The illusion. And so here we are seeing things a little bit amplified. I think.
Oh, there's a rocky outcrop on the right.
Sorry?
Oh, rock is coming.
Yeah.
Skonema Foliatum. And yeah, this one and it, and our community of encrusting sponges.
Yeah.
all settled on the area that is not covered by sediment. You see on the rock that you have a blackish pattern to the right.
Just very few are sitting on the sedimented
Yes.
part. Yeah, that's more for other organisms to occupy the sedimentary areas.
That looks like a Geodia parma to me.
Oh yeah, we haven't seen that yet.
You see?
The potato sponge.
That one. Yeah, so that's interesting. And Emigilius Arcoferus, sorry. So 2,200, you remember? On the first dives that the Geodia has always appeared at this. At this mark,
it's
and
starting
it was
to
exactly
get very, very distinct.
yeah.
Yeah, we had numerous dives now where at 2,200, at which depth we are also right now, we first began to see these sponges, these potato sponges of Geodia.
And we don't know if that happened on the unnamed because we never went that deep.
True, yeah.
So Geodias were there, but we don't know if that line of the 2,200 applies as well. But on the Boyd and on the shoals, apparently yes. Another Emigilius over there. And this one, can we stop for a while? No need to collect. Just want to have a look to that one.
Richard, can we slow down to 0.1, please?
Yeah, just to have a look. If possible.
Yeah, let's see if we can get out of the spot. Just pressed against it, so that's fine.
You see, Francis, it looks different than the other ones with the stem. This looks more like something that we have already seen. It's more irregular, the main body, the head.
Yeah, it looks like it's hanging its head or so.
I wonder if Felso is watching us, because he's the one that knows about these guys.
Let's check.
Could correct all the silly things that I say. Oops, sorry.
So I wonder, because it has the stem to be more further away from the sediment, but then it doesn't grow more upright, but it goes down towards the sediment. That doesn't seem so
Well, I guess
smart.
Can we stop this movement, please?
I guess that depends on the shape of the rock. I mean, it tries to go up, but the rock is covering. So yeah, that's what probably depends on where the currents are hitting. They always find the current and rearrange their body so they get the more food possible. And those things that are here on the rock, at the beginning I thought it was a kind of a weird Lysodendrorids, but it doesn't look like it, more fluffy and smaller. So I don't know what is that, never seen it so far.
But we've also seen that some Lysodendrorids are more fluffy than others, but this, seems too fluffy.
We don't know what
If
is, this.
you look at this, it doesn't look so much.
This seems
different.
like a Lysodendrorids to me.
Yeah.
But if you can see that the color and the morphology is different.
But
So
much.
Yeah. You know what? That's the thing. With the sponges, and you see all the little balls.
Yep, the calcareous
Those are
sponges.
calcareous sponges. So those are a different group, very separated from the other ones. These had the spicules made of calcium carbonate instead of silica. The very little ones with the stem.
Yeah,
So
which are more grayish.
yeah, Solvin took a very nice picture of one of those.
Oh yeah, I have to check it out.
Yeah.
Solvin is our underwater photographer on board, marine biologist, and he's doing in the lab also photos of the samples we take.
You can see super common, the calcareous sponge.
Yeah.
They're all over the rock. Do you want to see something? Nancy?
I'm good. Yeah.
Okay, we can move forward. Thank you.
It's a very green crust,
Yeah.
green-yellow crust.
And more Emicilius. The biggest one, or
Yeah.
big. Not that they are very big, but those kind of fun shape. And here we have another mysterious
Exactly.
stem. Very nice.
But this is like a really, really sponge-dominated community.
Yeah, deep sea, the sponge realm.
I mean, now more crinoids come into play, but
Probably there's not that much food here. So
you don't
sponges-
see on this wall also not many, not at all any at the moment, anemones.
No. Oh, we spotted a couple before.
Yeah,
Yeah. Crinoids.
but the sponges always
RV bridge.
Can we
manage.
resume movement 255 0.1, please?
Resuming movement 255 at 0.2.
0.1, please.
Copy 0.1.
I mean, those
Thank you.
little encrusting sponges, they don't need that much
there was
food.
a big sea spider, wasn't there?
I don't know.
Maybe too late, but looked like this or yellow-red one. But sorry, you.
No, I was
were
saying that these little sponges, they don't need that much food, the grass, because they may look kind of big, at least in the area that they cover. But if you measure how much tissue is in there, it's nothing. It's like half a gram. So the strategy then is to cover a lot of surface, so if there is not much food in the water, they can still capture some, because they have more surface exposed to the water and the food on the water. But they are not that big. They are just 0.5 grams. So this is not a place with a lot of food, probably. And that's why we find these kind of communities. It's oligotrophic, that's the word.
Nutrient poor water. Presumably.
Presumably, yeah, this is just guessing. It's still very diverse. You know? We take one of those rocks and we start finding more and more species.
Yeah. Also in the sediment there's a lot that we cannot see of worms and mollusks and bacteria, etc.
Yes.
All playing their roles in the nutrient cycling.
Nigeria. Yeah.
Oh yeah, more of them coming. We're also at about 2,200 still. What I also found interesting, what I think you told me the other day about the microbial dominated sponges, and the ones that are not, like the ones that are just made up to a large extent of their body mass out of, not of sponge, but of microbes.
So there are two types of sponges, and these are two categories that we have invented. They are not real categories in terms of phylogenetic terms, but some sponges, they have a lot of bacteria in their tissue. That means that if we take a piece of the tissue and we weight that piece of tissue, and then we remove somehow the bacteria and we weight the tissue again, we'll see that probably about half of the biomass is bacteria. And also in terms of diversity, there's a huge diversity of bacteria living inside one sponge. While there is another group of sponges that have much less amount and diversity of bacteria. So the first one are called HML sponges, and the second one are called low microbial abundance, so LMA sponges, so high microbial abundance and low microbial abundance sponges. And they play different roles in the ecosystems. The low microbial abundance sponges tend to eat larger food than the high microbial abundance sponges. We tend to filter a little bit more of water. So yeah, different roles and different strategies to survive. But for most of the sponges we don't know what they are because we haven't checked.
So you're checking.
Oh, 20.
Hey, fish. Whoops. So the thruster of the ROV that are used also for navigating, etc. They have some power, but luckily these sponges are very good at regenerating
No,
their body parts or their whole body. Yeah.
I don't think we haven't brought any sponge. Those,
Huh?
I don't think they. I think we don't have. We haven't broke any sponge now.
I saw a piece of Lesodendorix, our favorite, very popular sponge.
The ones with the stem, they are very strong.
They're very rooted.
Quite resilient to the roots, yeah.
How do they grow and attach themselves? Do they have mucus at the stem, or that serves as a glue?
Those are enzymes. So those are proteins that dig into the rock, dissolve the rock. So in the shallow waters, there are sponges that are adapted to grow inside rocks. And then you have, if you collect the rock, it's completely
Oh, the cook is coming.
halt. Good.
Hello, Molly.
Hello. What's up?
The chef that is tempering us with a lot of nice food every day, multiple times a day, is paying us a visit.
So we're still climbing, right? 2,171. Yeah.
At the transects we're doing at the moment.
So I think that we have more geodias. Yeah, you see it to the right. It's high. There is a really huge encrusting sponge over there.
Oh yeah, and also some green crusts
Yeah.
just around, competing with it.
That's Knipovich-Rückens to the left. is a polymastia, little polymastia.
Oh, the little ones.
Shrimp.
Yep.
That's a big shrimp, huh?
Yeah.
See how big is that one? Alright, 10 centimeters. She's really big. Now this is beautiful. Look, all these rocks.
Yeah, they have cool structures here. So the pinnacles and outcrops. And the organisms, the filter feeding and suspension feeding ones, they like it as well.
Shrimps, there's plenty of shrimps, you see?
They also get some nice food here. It's always funny how they like
they
walk
swim.
through the water.
Be careful, because
So the big ball with the stem, I've seen little ones. I don't know if it's the same, if it's something
If
different.
it's a baby or
Yeah, I'm tempted to. Well, this is too steep to stop, but
hard terrain.
a flatter or some kind of platform.
Yeah.
And maybe we can ask.
We can always
Can you
ask.
see this one? Yeah. And this one, they look different to me.
Yeah. And maybe that little one was next to the shrimp was also here. Little amphipods. Can we see them well like this?
Hemigilius sarcophagus again.
Oh, the fan-shaped one just below.
This one, when we collect it, it has a lot of mucus, and we are interested in sponges with mucus because we can study the natural products.
So very slimy ones. I just have to check that this
How it works.
it changed this screen.
Yeah.
Oh, now it's good. Yeah. Okay.
So with Paco, we collect some of these hemigilius in cold. We freeze them, and we will potentially study them in the University of Uppsala for new molecules with some potentials, like in biomedicine, for instance. Sponges are the most important group of marine animals in terms of how many new molecules we found per year. So they are like a big reservoir of resources as well for the humans.
0.2, please?
We can
Thank you.
be inspired in the structure of those molecules to then create new drugs. So
With
that's
antibacterial or antiviral,
exactly.
anti-cancerous.
Anti-cancerous, yeah, many
functions,
different
effects.
applications. So the biological diversity that we see here is matched with the chemical diversity, and this is also a reason to study these kind of environments, to put a name on the species, to collect some of them, and also to protect them.
Is this a little Jodia? Or is it a different
yeah, it
one?
looks like to me.
Little potato.
So you see the small one that I told you before, the one in the center? That looks different than this one. You see, it's more regular, it's more like a ball. I feel like there is this ball, and then there's a ball with the cylinder on the top.
Yep.
And then there is one that is just more irregular shape, like
That is-
cauliflower.
Yeah. Oh, cauliflower. No.
Yeah, cauliflower-like. So potentially I see 3 morphologies. You know, maybe it's all the same, but maybe not.
You have a little sketchbook for the different sponges? I saw you drawing the spicules at least today when you were sitting at the microscope to like
It
see
helps.
which.
I draw a lot when the samples come. So if we collect a rock with many rust, I try to draw the rock and then to take notes on it and to take a picture on that, because I want to match what I see on the video with what I have on my hands on the lab. Like that, then you can extrapolate the identifications with the videos. And you don't need to collect that much. Just collect once or twice, and then you can put a name on what you see with a little bit more of confidence.
So is this also a fan? Not a fan. A stem with a weird shape, like the one that we saw before. That was like melting down.
That one over there.
Oh, we see the cameras and the lights of our ROV one.
Yeah.
Some technical excursion. Oh.
So yeah, that looks more like the regular, like the cauliflower one that I was telling you before. You see?
Yeah.
It still has a stem.
Yeah, but it's like, it looks like melted.
Yeah, but it's not round. And then you have the ball-shaped one with the cylinder.
And there, below the more the cauliflower.
Yeah, so these are 2 different things, for sure. Let's see if I find the third one, the small ball. So it's 1, 2, and then there is potentially a third one.
And how do you call them? Just stem sponges?
Yeah, I mean, those are glass sponges. They are not my field. We've been using the name. There's something here. We've been using Amphidicella, Calophagus sometimes, but yeah, with not much confidence. At least from my part, I'm more of a demo sponge guy, which is all the rest you see. But those big ones with the stem, those are exactly glass sponges, and they are very different to the other ones. So you need to specialize only in that group in order to understand them.
It's always crazy on how much you can specialize. You can specialize on first marine biology, then deep sea biology, then sponges, and then glass sponges. Just endless.
Oh, that's beautiful.
Oh, hey, there we have a nice Ascanema.
So
And nice rock.
Gelias,
Oh, yeah, plenty.
Yeah, they really like the food. So I think that every time that we have a rocky outcrop, Gelias are there.
And a lot of the glass sponges. Oh yeah. Ilpout paying us a visit as well. Gathering some food.
Bridge RV.
The bridge.
Can we change the bearing to 250, please?
50 and 0.2, okay.
Thank you.
No, I thought this was a soft coral, but it was Asconema.
Those are really big. I think about it. So at some point we may try to collect one of those.
When the
see-
terrain is a bit easier.
Yeah, to see if we can send it to our colleague that is a specialist.
Is that colleague also in Uppsala or somewhere else?
No, so he was in Portugal, but at the moment I'm not sure if because it's from Brazil, I think. So I think it's back home. I'm not sure.
And we have a little jelly.
Jellyfish. No chance to stop in any of those places. Eh?
Well, stopping is one thing, collecting's different.
For collect.
But
For
we
collection.
can collect with the T4.
With the hand, you mean?
But we have to have space to open the trays. That's the only thing.
Being pushed
Yeah,
against.
I think it's going to be a little bit hard also with all the sediment coming then at us.
With the washes out.
No,
Yeah.
being pushed.
No, I'm not saying to put to stop now. Just
Yeah.
asking if it's okay to stop at some point.
This is one of the small ones.
Yeah, exactly.
Oh, there are more coming up. The small stem sponges.
So I don't know if those are baby whatever, the other one, or different species.
Bridge RV, can we change to 0.1 the speed, please?
6.1.
Unfortunately, the current is pushing us into the wall.
Yeah, let's carry on then.
Yeah, I never said to
Yeah.
stop it.
Also with the sediment, it's not so easy.
So yes.
Getting more.
There's a
I think-
stem there. You see that stem?
Yeah.
It's probably Lemisidius. Yeah.
With a crinoid on top.
Yeah, you see it from the side.
Yeah. Huh. Like a little palm tree. Out of two organisms. Yeah, more and more geodes. There. And we're now at
I think we
2128.
can land here if you want. We can. Where are the samples going to go?
To the bioboxes.
Yeah, which port or starboard?
The one you prefer.
If you want them in the starboard side, I can turn my head to port and we get it out. And if you want port side, I can turn my head to starboard to get it out.
You're saying because of these sponges?
Can be both. So the one that you prefer.
It doesn't matter where you want them, sort of thing. Because at the moment, I think there's one in the port
Starboard.
side aft,
Starboard.
and there's starboard. Okay, so if I turn the head this back port, turn to starboard side. So you tell me where you want me to rest. What
So
area?
we want to collect one of the balls.
One of these? Yeah.
One of those.
Okay.
Bridge RV, can we have a movement of 180 for 20 meters at 0.1 and then stop, please?
180, 20 meters at point 1, okay?
Thank you.
The one that is in front.
So Julio saw interesting sponges. that we want to look further on in the lab, what exactly they are
Cell.
and how they differ from each
We
other.
don't have
So we're coming now to a
hole.
halt, and it's a little bit tricky terrain because of the slope and all the fine sediment that is sitting on top of the rocks that is now being resuspended. But yeah, we'll try.
Yeah, yeah. Just taking some of your copy because it's in front of the RV.
So what do you think of this?
I'm not sure.
If it's a juvenile one or just a grown one of a different sort? Huh?
I'm not sure.
Yeah.
The pores of that would definitely look different.
They look different. Yeah.
And I mean, in addition also to its shape. But yeah, super large pores.
Yes. Yes.
Very spongy.
Can we remove the lasers for a second?
Sorry?
Can we turn off the lasers?
Turn off the lights, yeah.
Thank you.
Maybe on. No, too tight. No, okay. Sorry, can't get it. It's too risky.
Okay, that's fine.
The umbilical in front of the RV.
Yeah, no problem.
Yeah.
No problem.
The swell is a little bit big.
Mm-hmm. Yeah, we feel it as
No
well.
problem. No
Yeah.
problem. Yeah. Thank you.
No. If at first you
Maybe
don't see
if we change, if we turn over having
if you go
Start or we just try
in
to
the
play
port
it.
box,
Because
yeah.
probably at the top
on
it's
the,
also.
on the
Yes.
port box, it would be good to.
Yeah, but don't worry. We can
We
wait
can.
for later.
If we turn port, we have the umbilical behind us. If we, like this, we have on the front.
I would just say to move forward.
Okay.
Yeah.
So we're gonna move forward as the umbilical, the line with which the ROV is connected to the ship and all the swell is something we always have to look out for,
You pull it up.
and that makes it even more difficult at the moment to sample at this point. But we're pretty sure that we can find these sponges also more later on
That's it.
when the train is
Ship
more
is no
easy,
movement.
makes it more easy to
Shut
collect.
dry lab.
Go ahead, dry lab.
We're about to pause the dive stream in about 2 minutes so that we can go live on School Street.
The rocks.
Okay, thank you.
So yeah, we heard that the stream is now stopping soon. We're coming to an end because we're also doing some educational work with the school, for which we will also need the bandwidth. Yeah, but we've just found a interesting rock structure here. So we're gonna continue exploring here, and we will probably meet you later on in the next dive stream, and we can talk more about what we've encountered then, also what the lab processing, etc., has brought up. So thanks a lot for tuning in, and we hope to have you soon again.